Hello everyone, you are all welcome to lecture number 1248, and today's topic is the chemistry of indigo. Indigo is plant product, it is natural dye, very beautiful blue in color, and this is one of the oldest natural dye used by people to color clothes and so many other materials, and it is isolated from one of the tropical plant, that is Indigofera species. And this Indigofera plant, the one which is known as Indigofera nautica, is actually a rich source of this blue dye, and it is also having some medicinal importance. It is powerful anti-inflammatory, and this natural dye is heterocyclic. It contains aromatic ring and 5-member aliphatic ring, and it is made up of actually 2 units of isatin molecule, and that one I will be discussing on the basis of some reaction when we are discussing the property of indigo, that what type of structures and carbon skeletons are there in indigo molecule. It is also used in some drug as well as pharmaceutical industries for different purpose, like antimicrobial, antibacterial purposes. But mostly the indigo dye is popularly known worldwide as a natural dye, and it is found in the form of glucoside. That's why, because of its glycosidic nature, it is water soluble. So it is found in the form of glucoside, where glucose molecule is linked with one of the aromatic ring. So here I have written the molecular formula of indigo. Actually, when I write indigo, it is the mixture of indigo. It is the mixture of different type of indigotin molecules, but all those things are having related structure. That's why the common name is indigo. Otherwise, the molecule, if we are very specific, it is indigotin. And this indigotin is called indigo, which has molecular formula C16, H10, O2 and N2. And when we are checking its structure, and once it is subjected to oxidation, it is broken down into isatin molecule, and this is isatin. And isatin is nothing but it is indole, and where at 2 and 3 position there is keto group. That's why it is called indole 2,3 dione, and indole 2,3 dione is called isatin, and the final product it gives, that is isatin. So it clearly confirms that indigo dye, or we can call it indigotin, is made up of 2 molecules of isatin, and 2 molecules of isatin can be in these 2 structures. And here we have to see its geometry, which one will be more stable. So that I will be discussing on the basis of some of the reactions later on, and on that basis we will say that the structure 1, that is A, is the stable structure of indigotin or indigo, because it is active and it shows geometrical isomerism Geometrical isomerism means where the free rotation of these two portions of isatin is not possible. It is arranged either in cis or trans form. That's why the restriction of rotation is there, and in that case, the structure A is correct structure for indigo dye. This is the INDIGOFERA plant species, which is very rich source of blue indigo dye, and this plant is economically very important because of its high content of blue dye. This is the image of blue dye isolated from INDIGOFERA plant in the form of glucoside and is ready for use. So in such type of big bundles and packet, this indigo dye is sold in the market, and it is really a very important economic source to those people who are extracting indigo dye from plant sources. So blue indigo dye, here number one, is an organic compound with distinctive blue color. It's very deep blue color. It is already shown to you how the big packet and bundles are sold, and very beautiful color plant of INDIGOFERA is also shown, and the leaves of INDIGOFERA plant is very rich source of this natural dye known as indigo. Indigo is a natural dye extracted from the leaves of INDIGOFERA genus, and the very particular plant which are used for the extraction of this indigo dye is INDIGOFERA tinctoria. And this indigo dye, this plant is commonly grown throughout the world, especially in tropical countries like India, Bangladesh, Sri Lanka, and also in some of the European countries nowadays, because now it is global source of natural dye. It has lot of economic importance due to its beautiful blue dye stuff. So here number two, its destructive distillation means when indigo is subjected to destructive distillation, it gives aniline, and getting aniline as one of the final product clearly indicates that indigo blue dye nucleus is having aromatic benzene ring. If aromatic benzene ring is not there, we cannot get aniline. Number three, its fusion with alkali affords either anthranilic acid or aniline, again confirming that indigo or indigotin blue dye is having aromatic benzene nucleus. Number four, when it is subjected to oxidation in presence of nitric sulfuric acid, we are getting two products, that is two molecules of isatin, and that is given here, and already the structure of everything I have explained in the beginning, that is C16 H10 O2N2 plus O2 means it is treated with a strong oxidizing agent like concentrated nitric acid, and then it gives two molecules of isatin, that is C8H5L, and this reaction clearly shows that indigo dye is made up of two molecules of isatin A and B, and the structure of A and B is given. Now we have to prove which structure is correct on the basis of its geometry. So this oxidation reaction shows indigo is made up of two molecules of isatin linked together and assigned together A and B structures. Number 5. The treatment of indigo with dilute alkali gives anthranilic acid and indoxyl to aldehyde. So here once indigo dye, that is C16H10O2N2, is treated with alkali, you will get this acid, and this acid is called anthranilic acid, and plus one aldehyde substituted product that is called indoxyl. So the above reaction, the formation of two products can be explained only by the structure A and B, which is already given. The structure A is capable of showing geometrical isomerism, means where the carbon-carbon double bond rotation is restricted, and it can be either in the cis form or trans form. But in the case of B structure, geometrical isomerism is not possible, and since this compound is optically active, it is showing geometrical isomerism. It means clearly the structure A is correct structure for indigo dye. So with this, I will stop. Thank you very much for watching the lecture.